Good morning, everybody. Welcome to the webinar series on wastewater surveillance for SARS-CoV-2 and emerging health threats. Kyle will do an introductory set of slides in a little bit. We'll wait a minute or so.
While we let people join, I'll also throw up these QR codes and I'll also put the links in the chat. If you're not registered for our mailing list, you can sign up to keep updated on our webinars. And also, if you would like to nominate a speaker for a future webinar, I'll leave this up for a second. All right, just to keep things moving, we have a slate of great speakers today. First up will be Krista Wigginton and Abigail Monahan from University of Michigan, talking about something I'm personally very interested in, tracking viral shedding a temporal time set. So very excited to hear about that. And then we'll have Noor El Ul Huda from University of Antwerp talking about tracking hepatitis E using wastewater surveillance. So also very excited about hearing that. We also have an update. Allie and I are planning on continuing the seminar series next year. We'll take the summer off and start with next academic year, which will be September 4th. So it'll continue to be the first Friday of each month. But we have a clean slate of speakers. There's a few folks that have demonstrated interest, but if you would like to nominate yourself or other speakers, I'll throw this QR code quickly up, and then I'll also drop it in the chat. So with that, I'll invite Krista and Abigail to begin speaking once they have the opportunity or once they are ready.
Okay, can you hear me? All right,
We
great.
can, yep.
Great. Thanks, Kyle. Thanks, Allie. Thanks for inviting us here today. So I'm Krista Wigginton. I'm at the University of Michigan. I'm part of the Wigginton Eisenberg wastewater monitoring team. Marissa Eisenberg is the co-PI on that project, and she's in epidemiology. And so we do monitoring for a number of facilities in Southeast Michigan. Um, and so I want to talk today about a fairly new project we have with some preliminary data on quantifying viral pathogens in urine and stool through kind of trajectories in community members. Abigail Monahan is a PhD student in my group and the lead PhD student on this project. Betsy Saltzman is a technician. Abigail's going to talk in a little bit and present some of the preliminary work, but I want to give, and I know Abigail's going to bring Betsy up again, but she's been a core member of this team and pulling this cohort together and organizing it. So shout out to Betsy and the other co-authors, and of course, Marissa, my co-PI. So I'll get right into it. I just wanted, with the next slide, I don't need to talk about wastewater monitoring, but that in this case, we're really kind of focused on the ability to predict or use wastewater, not for trends, not for presence absence, but for trying to pull out some epidemiological parameters, things like absolute burden in a community. And so that's kind of the application we're talking about, is taking the quantitative measurements and then inferring something about the epidemiology in the community. So that's one way, right? Like looking at wastewater and hopefully looking back at the community and trying to understand how many people are infected and other things about the transmission. But there's another way to think about this too, is like a forward mechanism. Can we predict based on how many people are sick and what the shedding looks like, what worst case scenarios we might see in wastewater? Because when we're thinking about, especially in water reuse, potable water reuse, when we want to understand the amount of viral log reductions or pathogen log reductions we need to achieve, we're often limited with measurements that have been made in wastewater as the starting kind of concentration. If we understood really well how people shed, if we could model illnesses in a community, understand something about how many people will get infected and what that would result in wastewater concentrations, we'd have more confidence over the starting material that we're working with potable reuse, and be able to better inform the kind of guidelines that we have on how much virus must get removed through wastewater treatment and advanced water treatment. So kind of thinking about this both ways. For either way, in order to go from absolute abundances in wastewater to understanding about burden in a community, if we want to go that way, or if we want to go backwards, or I guess it depends what we're talking about, forward or backwards. If we want to predict worst case concentrations in wastewater for maybe more of the GI type illnesses, we need to have a pretty good understanding of what shedding looks like. We need to understand how much people shed, at what stage in their illness, and have really well-resolved trajectories for all the illnesses that we're interested in, so we have confidence in that transition between the community infections and what we observe in wastewater. So that's the motivation for this work. This is just a few examples of papers or reports that have tried to do this, have tried to use wastewater to get at some kind of epidemiological value, or in the last case, it was a report that we had and where we were using fecal shedding to try to predict wastewater concentrations for water reuse regulations. Often these efforts where they're modeling with wastewater data or modeling with shedding data, they note that the lack of shedding data is really kind of limiting the ability to do this and creating a lot of uncertainty in the values that they predict. And so I think, maybe some of you have tried to do this on this call, but we really are, for a lot of pathogens, there isn't a lot on fecal or urine shedding. Part of that is because when we're talking about respiratory pathogens, there maybe wasn't, before wastewater epidemiology, much of a reason to understand how many gene copies are being shed at what point in infection. So, but since wastewater-based epidemiology has grown in use, I think this has become a pressing need. So there's different ways to do this. I'm going to talk about some of the study design. So one would be a challenge study where they actually infect patients with a pathogen and see if they are infected. They often keep them in the hospital and are taking samples from that person over time and quantifying how much, whether they're shedding or not, and sometimes quantifying how much. There's limitations for this in terms of the applications that we want. This doesn't mean that those aren't valuable for many reasons, but some of the limitations with the resulting data is it doesn't often get the pre-existing immunity. It often is very focused on one pathogen, so you don't really see anything with co-infections. And they're hard to scale. This is a time-intensive effort. Not everyone gets infected, and you're limited with how much you can get in the end. The other one I'll mention is a clinical study. So that would be people show up at a clinic and they get tested for a pathogen and they test positive, and then they get enrolled into a study and they start taking samples. So this limits, or the limitations of this for this application is it's limited to people who show up at the clinic. So they're symptomatic and sick enough to show up at the clinic. They tend to have certain demographics or concentrated in certain demographics. It's limited in how much they're actually representing the public. One other one is a family cohort study. So similar to what I just mentioned, somebody shows up at a clinic, tests positive for an illness. They enroll in the study. They can sometimes enroll their whole family and start monitoring their family members. And then the idea is some of their family members are probably going to become infected. And then you do get some earlier infection data from those individuals. There's still some limitations with that. It's a lot of effort to do these family cohort studies. You're often just looking at one or limited pathogens, and you do get some limitations in the demographics of the people that show up to the clinic. We were involved in a project like this, ran by Daniel Kelly at UCSF. Peter Arts was a PhD student in my group, and he looked at the shedding of SARS-CoV-2 in this family cohort study. It was a really valuable data set, but it was a lot of work to get those samples. And also it's hard to get all of those family members to continue to give fecal samples. Getting fecal samples is not often a fun thing for people to do. And so we struggled to get a lot of participants to give us a full set of samples. So some of the things that are often not captured in these studies is this pre-symptom onset. So the curve how much people are shedding before they actually start to feel sick and then before they show up in the clinic. And that's important data if we want to have good models that kind of estimate what the abundance should be in wastewater over time. Another thing is some of these pathogens, they're shed for a very long time. And a lot of these studies are designed just to, have people in the hospital for a week or limited with how long they might enroll the participants to give samples. And so often the curves are missing these long-term shedding that might happen after the symptoms have resolved. Some other issues there's data out there for lots of pathogens, limited data, but there is some, but often they weren't designed with, the ways that we want to use this data. And so, for example, sometimes they report, a lot of these previous studies, they report their concentrations in CT values as opposed to absolute abundances. And without a standard curve, you can't use those CT values very effectively to predict absolute abundances in wastewater. Oftentimes with stool, they'll take a swab and they'll measure the swab, or they'll just take the stool and extract from the stool and measure it based on the mass of the stool. People can shed very different types of stool, like with different water contents. And so it's rarely have these studies normalize this data to dry weight of fecal shedding, which is important because that's much more consistent person to person than the wet weight of stool. So if we want to scale that up to how much someone's shedding over a full day, having the dry weight is a much more quantitatively robust measurement. And then a lot of studies just look for whether it's there or not, which can be valuable. Like, okay, yes, a person sheds this pathogen, but that's obviously not going to be very helpful when we go out to communities and look at what their quantities mean. So with all that in mind, there's kind of this ideal data that we'd have for wastewater applications, right? We'd want populations that are representative. We don't really want really niche populations within the sewer shed because they might have wildly different shedding characteristics than the general population. We want to get those full trajectories, both pre-symptom onset and these tails that happen after the infection has really, they're no longer testing positive in their with normal clinical tests. We really want these concentrations to be externally valid. And by that, I mean that if we report 50 gene copies per milligram of dry weight, someone else would measure that too, and they'd be able to use that data to inform the wastewater measurements that they're making. And then, of course, ideally, a lot of these studies are very specific, studying influenza A virus under these kind of strains. It would be great if we could monitor for lots of pathogens at once and get a lot of curves at the same time. And so that leads to the motivation of our study, which is to generate the first of its kind of viral shedding for many pathogens in the general public of our community. So now I'm going to pass it to PhD student Abigail Monahan, and she's going to report some of the results we have and a little bit more about the study design.
Awesome. Thanks, Krista. All right, ready for the next slide. Okay. We are taking a multi-phase approach to this research objective, beginning with designing a multi-year cohort study. Then we will design a multiplex ddPCR assays to quantify multiple respiratory viruses. And finally, we will quantify virus concentrations in those cohort samples. We are focusing our quantification on respiratory viruses because of the public health applications for respiratory viruses, and also because a lot of these pathogens are also monitored by our wastewater monitoring efforts that Krista mentioned in the beginning. So specifically, where we're looking for human rhinovirus, adenovirus, flu A, flu B, COVID, and RSV. In order to design this study, we determine the ideal cohort size based on infection attack rates so that we can anticipate the number of curves that we will be able to quantify based on that infection attack rate. And so we have set a goal for a 100 participant cohort with the belief that it will result in quantifying around 95 rhinovirus curves, 25 adenovirus curves, and so on.
Sorry, Abigail, I'm trying to forward here.
That's okay.
There we go.
We designed our cohort study, which we are calling the Shedding Community Pathogen Evaluation, or the SCOPE study, to quantify well-resolved shedding curves with the objectives Krista mentioned in the previous section. So we begin by enrolling participants from the general population over the age of 18 years old who meet our study requirements. They collect samples weekly for 16 weeks. They collect a stool and urine sample and take a rapid test for COVID, flu A, and flu B. And if a participant has 2 or more influenza-like illness symptoms, which they report in weekly symptom monitoring, or a positive rapid test, they enter what we call an enhanced sampling period, where they collect samples daily for a week, which can proceed into 2 weeks if their symptoms or their positive test result persist. And then following that enhanced sampling period, they return to the regular weekly sampling cadence. Once the samples are collected, they can be kept in the participant's refrigerator for up to a week before delivering them to us, where we aliquot the stool and urine samples, we quantify the dry mass of the stool samples, and we stabilize our stool and urine samples with DNA RNA shield. After samples are aliquoted, they're stored at -80 until we're ready for that analysis. We begin by pooling samples into groups of 4 and then performing total nucleic extraction with the Kingfisher and the Magmax viral pathogen 2 kit. We store extracted nucleic acids at 4 degrees C for up to 48 hours before doing ddPCR on both undiluted and samples at a 10-fold dilution to account for inhibitors in stool samples particularly. And then samples that are positive, pooled samples that are positive, we then run the individual 4 samples in that pool for absolute quantification. The reason we have taken this pooling approach is because at this moment in time, after 2 years of this cohort study, we have over 1,000 bank samples, and we anticipate that the majority of those do not contain virus because they're collected by regular members of the general population that are healthy at the time of sample collection. And so taking this pooling approach allows us to do this efficient screening in order to determine which subset of samples we should go back and analyze for absolute quantification. We designed 2 multiplex ddPCR assays, the first of which tests for flu B, SARS-CoV-2, RSV, and bovine RSV, which is our extraction control, and the second tests for flu A, rhinovirus, and adenovirus. Samples that are positive for adenovirus and undergo an additional ddPCR assay so that we can identify the species and strain. On every ddPCR plate, we run an extraction positive control with bovine RSV and DNA RNA shield, the extraction negative control with DNA RNA shield, non-template control, and for positive controls for our targets, we are using gene fragments. At the end of the first 2 years of running this cohort study, we have assembled a cohort of 46 individuals that is fairly representative of the Ann Arbor sewershed in terms of sex, age, and race and ethnicity. We recruited 26 individuals in the first season and 34 individuals in the second season, 14 of which were returning from the first season. And so that's why we have an N of 46, but a total of 60 participations. Over the course of those 2 seasons, we had 19 enhanced sampling events, 10 in the first season and 9 in the second season, 3 of which were triggered by a positive influenza A test, 1 triggered by a positive flu B test, 1 triggered by a positive COVID test, and the remaining 14 triggered by folks experiencing 2 or more influenza-like illness symptoms. We had 52 of our 60 participants, or 87% of our participants, submit 16 plus weeks of samples, which we consider 100% completion of the study. And we attribute that to our participant retention strategies that we have built into the study, specifically allowing participants the flexibility of storing samples until it's convenient for up to 5 days, until it's convenient for them to deliver their samples. Their samples can be delivered via handoff or as is convenient for them within working hours in the lockers here with a locker code that they have. We have a generous compensation scheme, which includes weekly payment for their stool and urine sample collection and positive and rapid test, as well as bonuses for every 4 weeks of participation in the study, and our regular study communications and check-ins, thanks to Betsy Salzman, our project leader, who is a phenomenal study coordinator, as is evidenced by the communication we have continued receiving from participants over both seasons, thanking us for the opportunity to collect stool samples in their house, which is an insane thing for 16 weeks in the winter when it's freezing cold. We just have had phenomenal participation from our community, and that is in huge part due to Betsy's efforts. We have done a subset, we have analyzed a subset of samples, specifically samples collected in the 2024-2025 season. And you are looking at some of the initial results of pooled samples. Of the 117 pooled samples analyzed, representing a little over 450 stool samples, we had 20 positive pools. And the figure you're looking at on the right here is the number of positive pools that were positive for one or more viruses, and the viruses they were positive for is on the x-axis. What I'd like to highlight here is that 13 positive pools, so more than half of the positive pools, contained only regular weekly samples collected from participants. They were not collected during an enhanced sampling period, and I think that speaks to the strength of the design of the study to capture so much of this asymptomatic infection. We'd like to show some of the preliminary curves that we have quantified, beginning with influenza A shedding trajectory from a participant that had a positive rapid test. They had a positive rapid test on February 13th, 2025, as would be predicted by the peak of that shedding. But I think this figure also elucidates that shows that we are able to quantify some of that pre-symptomatic, pre-positive test shedding and the full shedding trajectory through the cessation of shedding. We also have a shedding curve of a participant that was positive for adenovirus species C. And what's interesting here is that the participant was asymptomatic. They were not in an enhanced sampling period. And so they were doing weekly sample collection, and yet we were still able to get these shedding trajectories. Our ongoing work is to continue analyzing samples from the 2025-2026 season, both stool and urine samples. We have the majority of samples collected are these weekly samples, with a little over 250 of the enhanced samples. We have analyzed a little less than half of our stool samples, but still have all of our urine samples to analyze. And we collected 935 samples from participants in that first season, a little over 1,100 in the second season. And in order to reach our cohort goal of 100 participants, we anticipate needing to collect about 1,400 samples next season. Future work includes expanding to our cohort to include folks under the age of 18, expanding our symptom reporting to capture instances of gastrointestinal illness, such as norovirus, and then applying these resulting curves and modeling efforts to estimate community burden. And these fecal shedding curves will allow us to translate wastewater measurements for both wastewater-based epidemiology purposes, where we're able to predict community burden, and to establish evidence-based log reduction value targets for advanced treatment systems. And we'd like to acknowledge the Wigginton group, Marissa Eisenberg and Betsy Salzman, GRFP Flu Lab, and Michigan. Thank you.
Great. Thank you, Krista. Thank you, Abigail. Very exciting presentation. So just a reminder for folks, if you have questions, drop them in the Q&A box. It should be on the bottom menu bar of your screen. We already have a few questions there. So the first one I'll ask is from Prabhu Gandhar. asking that shedding can vary within individuals year to year, for example, due to immunity or, the SARS-CoV-2 shedding may decline with subsequent seasons. Um, how would you think about accounting for such factors and able to generalize to other settings, seasons beyond this cohort? And either Krista or Abigail can answer.
Yeah, I can start, and if Abigail, you want to add on to it, that's fine. So I think we wondered in this design, because people want to come back and participate again, but we were torn, like how much to allow repeated participants versus how much to try to reach out and get new participants. And so I don't think we're going to have the sample size to answer that kind of question, but we're hoping, I think, Abigail, what did you say? Around 15 of the second year had also participated the first year.
Yes, yep.
And I don't think we have the data yet to say whether or not any of those people's experienced the same pathogen in 2 years. My guess is that our numbers are not going to be high enough. Like I don't know if we'll see any, let alone if we see any, what that really means. But it's a good question, and maybe, in the future, as we get, not we, but as the scientific community gets thousands and thousands of these curves with a little bit of information of year-to-year illnesses, we can start to answer that. Abigail, anything?
No,
Okay.
nope.
Great. From Colleen Naughton, sort of an operational question. When they are sick, are they allowed to have a relative drop off their samples?
Yes, yeah, we allow for anybody to drop off. Allowing participants to keep samples in their fridge for up to 5 days is for a regular week, but we absolutely make exceptions when folks are ill, just to prevent them potentially infecting others in the community. And yeah, allow them to recover before delivering those samples. So that delivery is a little bit more flexible if they're in an enhanced sampling period.
Great. How did you quantify the stool dry mass?
Dry mass is quantified by massing the initial wet weight of the stool and then drying the stool sample on a heating block at 99 degrees for 24 hours, and then requantifying the stool when we know it's completely dry, and then getting that dry mass fraction, which we use as an element of elucidating that final concentration.
Right. How do you imagine a public health department that currently monitors respiratory virus trends via wastewater would be able to use this resolved shedding curve data?
I'll take a crack at that, Abigail. I think, yeah, I don't know if the public health agencies will directly use this data, but the tools that get developed on how public health agencies can use the wastewater data could incorporate this, right? So if they're trying to figure out these conversions and like estimate absolute abundance or absolute burden in a community, or things about how easily pathogens are transmitted from person to person. I think that's where the models that are built that try to get at those values will use this shedding data, and then that gets passed to the public health agencies. So they'll have more information about what's really happening in the community with this kind of embedded in the analysis. If that makes sense.
Yeah, makes sense. And then the last question on a sort of a Q&A. Did you ask any questions from the participants about their vaccination status, and are you planning to consider that in your analysis?
Yeah, in our initial intake study, we ask about vaccination status as well as some dietary questions. And then the weekly symptom reporting asks about symptoms they're experiencing specifically.
Great. Well, thank you, Krista and Abigail. This is clearly very important data. So with that, we'll move on to our second talk. Noor, if you wanted to turn on your camera and share your slides when you're ready. And just for everyone else, as we transition to this second talk, just a reminder to put any questions you might have for Noor in the Q&A as she goes through her presentation.
Can you see my slides?
We can see them, but it's your screen. It's not in sort of presentation full screen mode.
Yep.
Great. This looks great. Thank you.
Hello everyone, I am Noor, a PhD student in University of Antwerp in the Laboratory of Microbiology, Parasitology and Hygiene. Today I will be presenting on hepatitis E virus. This is a part of a chapter in my PhD thesis. In the whole presentation, I will go through the background, what is the hepatitis E problem, the study design and method we have used, the key findings, conclusion, and future perspective. And this presentation is based on the manuscript that is already submitted, and we are waiting for the reviewers to comment. So first, what is hepatitis E virus? I will go through a little story behind the hepatitis E virus. So around in 1978 or 9, there were pregnant women were reporting jaundice in the area in Kashmir. And the scientists were looking into their blood samples, but they couldn't find diagnose it because it was negative for hepatitis A virus. So there was something that was there, but it was not hepatitis A. And then later, after a decade, a scientist, a very brave scientist, Michael Balan, he actually ingested some stool samples from some soldiers who were suffering from the similar jaundice disease. And then he found a kind of different virus in microscopy. And later in around 90s, it was named as hepatitis E virus. So it's a positive sense, single-stranded RNA virus. And the interesting thing about it that it exists as a quasi-enveloped and non-enveloped form. Why it's interesting? Because then because of these characteristics, its instability and transmission routes are different. It's in quasi-enveloped form, is in blood, and that it can transfer with blood transfusion. And when it's in outside in non-enveloped form, it can transfer through the fecal oral route. So first, it was limited to the low and middle income countries, and then later on, there were few cases started coming in Europe and in America and in Japan. So then the people were suffering from the jaundice, and they were not positive for hepatitis A, of course, and they had no travel history. So then it was not anymore the disease of the low and middle income countries. So it has 8 genotypes so far. These 4 are important, genotype 1, 2, and 3, and 4. So the 1 and 2 is in low and middle income countries, and the source is by drinking contaminated water. And the host is mostly humans, and it's up to 30% mortality in pregnant women as well. And it's there in endemic. Sometimes there are outbreaks, but usually it's endemic in those countries. But in high-income countries like in Europe, America, and Asia, its transmission is primarily zoonotic by eating undercooked pork and raw meat from boar and venison. Its host range is very extensive, and it's still animals are adding in it, and it's usually asymptomatic, but in immunocompromised individual, it can cause chronic hepatitis. So hepatitis E virus is actually, as I said, is emerging in worldwide and Europe. So according to WHO, there are 20 million hepatitis infection per year, but only 3.3 million are symptomatic cases. And in a European Center for Disease Prevention and Control, in a report, they showed that there is a 10-fold increase from the cases from 2005 to 2015. In most cases, are asymptomatic, and clinical reporting is really underestimate the true burden here. So why we decided to work on hepatitis E virus. So there was a study in Belgium where they surveyed serum bank from year 2006 and 2014. And by looking into the serum bank, they estimated some yearly hepatitis E virus infection. They estimated there should be 54,000 yearly cases in Belgium. But we have National Reference Center for Hepatitis E, where all the cases from the Belgium, they are reported from some laboratories as well and some from clinicians as well. So they reported that there is between 25 to 117 cases per year, and most of the cases are from genotype 3. But we saw in the previous paper that the estimation is 54,000 cases. But of course, most of the cases are asymptomatic, so we don't know what is the true burden of the disease in the population. So why hepatitis E genotype 3? That we saw that already in NRC cases, 92% from the genotype 3. And also there was a study which was done in Belgium on the pork meat products, and it also they also saw that ready-to-eat pork products and the liver pate, they all have the hepatitis E virus infected, but it's genotype 3. And pig and human hepatitis was also phylogenetically very closely related. So it confirmed that the hepatitis E virus circulating, genotype 3 circulating in Belgium, the main transmission route is by eating undercooked pork products. So here, as we have seen before, that most of the hepatitis E cases were asymptomatic. So there, the wastewater-based epidemiology plays a role, because for clinical and hospital surveillance, only people who are severely ill and who show symptom goes, and then it is recorded in the National Reference Center. But the wastewater, the people who have show symptom or asymptomatic, everyone goes, because everyone used the toilet, and you all know the process after that the wastewater goes to the wastewater treatment plants, and from there it's collected and then sent to the lab for the analysis. So in this case, for hepatitis E virus, the wastewater-based epidemiology was very useful, because it could show the true community level, which the other clinical surveillance and hospital surveillance misses. So wastewater-based epidemiology does not wait for people to get sick. It catches the signal in the sewer first. So we already have an established wastewater system in the Belgium. We have 30 wastewater treatment plants that are used for the surveillance, and they collect influence sample over a 24-hour period, and it covered the 38% of the Belgian population. But this, they have the dashboard, but the diseases they upload there is limited to SARS-CoV, influenza, and RSV. But as the benefit of the wastewater, that one sample can serve for many diseases, we use those samples to also detect hepatitis E virus in our lab. So we did a surveillance study for 2 years, 2022 and 2023. First, we did a pilot study in 2022, where we took only 4 wastewater treatment plants from one Antwerp province, and the sites were Antwerp North, South, Deurne, and Turnhout. And then we expanded this to national network by including 6 provinces in 13 wastewater treatment plants, and it represented 23% of the Belgian population. So the samples we got, they are actually collected through the automated samples. So they are a composite sample. They are the influent water comes and stored every 10 minutes, and then it got collected next day. And then the samples are sent to the lab at 4 degrees. So there are autosampler are installed in the multiple strategic location, and the samples we got there were also from the similar automated samplers. So this is the sample processing workflow. I will go through it quickly. So as I said, that we were receiving a composite influence sample. So we did first remove the large sludge things from our sample by centrifugation. Then we used the Centricon plus 70 filter with 100 kilodalton molecular weight cutoff. It has we did two centrifugation. First, we collected the filtrate, which is for the size smaller than 100 kilodalton. They were filtrate, and the leftover virus on the filter was again centrifuged to collect the concentrate. That concentrate, we did the total nucleic acid extraction with the Maxwell, and then to quantification, we have used the Quasity digital PCR. And for normalization, we have used the paper mild motile virus as a fecal proxy that is very stable in human feces, and it accounts for if there is a rainfall and a population movement. And then we calculated the ratio, and we also performed the limit of detection on PMMOV, and the samples that were showing values less than 125 copies per ml were excluded. So the dPCR gave us the values in copies per microliter, but we taken into account all the dilutions we did while extraction and concentration, and we had the results in copies per ml for both our hepatitis E genotype 3 and also PMMOV. Now we'll go through the results. So first I will show you what we have seen in 2022, where we did 4 wastewater treatment plants. So in the first graph, we see that there's sporadic increase. Also, there's a baseline. And in the graph C, which is in the bottom, we have PMMOV, and in the middle, we have hepatitis E and PMMOV ratio. So in the middle, we see that it's become a little bit more stable. Then we aggregated all this, the results of 4 provinces into the one graph. Here it's not aggregated to show that clearly that we have some sporadic peaks in our graphs. And then we aggregated them to see what is actually happening in the province. So we could see that in somewhere around summer in week 19, we have a peak, and then also a little into the somewhere around November, and it's mostly because of, I think so, the one station. And then if we move to the bigger wastewater stations, where this graph represent actually the 22% of the population. Here we aggregated all the provinces, 13 wastewater treatment plants, to see the results. So we could see that again in the middle of the year, but not exactly the same we have seen in 2022. We have peak, and again in the November we have peak. Around autumn time we had peak. So then we did also a spatial analysis, and we divided all our provinces and tried to look if they are different. So we could see that there were spatial heterogeneity. Like in Antwerp, we could see that we have it's a biphasic. We have somewhere around 28 to 32 a peak, and then again in the end of the year we had peak. In Brussels, we have baseline. There was nothing we could say about it. And then East Flanders, we also could see a peak around week 28, but it was short and narrow, and quickly coming back. And then Flemish Brabant, we saw the abrupt late surge in autumn, the most distinct pattern we could see here in all our provinces. And then in Limburg, it was low amplitude, recurrent, never strong, not clear. And also in West Flanders, they were broad, but then after was first elevation, it sustained for some time from spring to autumn. And then we compared the same wastewater stations we had in 2022 and which we have in 2023. The thing we could see that in 2023, the concentration for the hepatitis E was low. Why? This cannot explain it. Maybe post-COVID period or something, but to explain it more, we need to do more years to know why in 1 year in the similar wastewater station the concentration was high and the concentration was low. We also tried to compare it with the data we have from National Reference Center. So suppose these two graphs, they are in the graph 1, which is all the cases reported in the Belgium in the similar year, in the similar weeks, is plotted against the wastewater from the Antwerp province. We couldn't see any correlation here in the similar in Flanders. So Flanders includes Antwerp and other provinces as well, but it's not the whole Belgium. So Belgium is actually divided into Flanders, Brussels, and Wallonie. So Flanders is where all our other wastewater treatment were tested. So we compared it with Flanders as well, but still we cannot see any correlation. And also the number of cases were very low. You could see they are maximum 7. Again, we did for 2023 the similar, as we had more stations, so we tried to compare it with all the NRC data. But again, we cannot compare it with the wastewater and the whole cases reported in the Belgium. We again tried to divide the data from the wastewater into the Flanders and Brussels, because we had the results from wastewater and also the cases from the Flanders and Brussels. But again, we don't see any correlation between the wastewater and the clinical cases. So the wastewater signals, 98 to 100% detection rate, year-round circulation, and it can be quantified. Clinical notification we have seen, there are very sparse, seasonal, severely underrepresented, only hospitalized or symptomatic cases. So implication is that wastewater captures the true community level burden, then that clinical data miss here. So in conclusion, can say that hepatitis E virus genotype 3 circulate widely and continuously in Belgian community, and there is a sustained year-round presence detected more than 90% of all sampling, even across all the sites. There were summer peaks, not exactly the similar month, and there was spatial heterogeneity across the provinces as well. There were no temporal correlation with clinical notification, which we expect because most of the cases are asymptomatic. That's why wastewater-based epidemiology is useful, because it can complement and can show the true population level within the One Health framework. The future perspective is that we could do the metagenomic sequencing that would enable the strain level tracking and source attribution, identifying which circulation subtype are driving wastewater signal. Multi-year surveillance is essential to distinguish year-specific fluctuation, because now we see that in the 1 year there were peaks are in the summer, but 1 year there was a little earlier and there's a peak later. So to found a genuine long-term seasonality, we should do more years. And then we could also try to link wastewater data to food chain monitoring and animal reservoir surveillance, particularly in pig farming and hunting areas. It would place these findings within a true One Health framework. And in the end, I would like to acknowledge all the contributors. So the study was done in collaboration with Cienzano and also from the University Hospital, from Professor Thomas Monahan, and also my promoter, Professor Peter Gilpute. And I would also like to thank all my team members for their technical support. And thank you for giving me opportunity to present my work here. Thank you.
Thank you so much, Noor, for the presentation. We have a bunch of questions, so I will go through and ask them. Super interesting presentation. I didn't know about the history of hepatitis E, so I was interested to learn about that and also your data. So I guess the first question that we have here is, you presented in your introduction some of the data on pork products in the market that had hepatitis E virus. And there's a question of whether do you remember if those studies reporting that did molecular measurements or actually infectious hepatitis E? Like when they were screening the meat products, was it a PCR assay or not? Do you remember from those papers?
Not really, but no, I don't remember. I have the data from that they detected it, but I'm not sure about it, that if they did the PCR, but most likely they did the PCR.
Okay, yeah, because that's usually easier.
Mm.
Given that there's concentrations detectable in pork products, do you
Mm.
think that pork products like put down the drain or someone's garbage disposal, which everybody in the US has a garbage disposal? I don't know if that's common in Belgium or not. Are there ways that pork could end up in the sewer system other than through consumption? Or is there any possibility that the RNA that you're detecting from hepatitis E is from like a pork product, like if it just passed through the human body like PMMOV does?
Mm-hmm. Yeah, there is a possibility that if they're taking the products and it passed without infecting or without going into the blood, but then it will be very minor as compared to
Mm,
what we were detecting, because of course it's not multiplying into the body. So there will be there for sure, but it should be very minor.
hmm. Okay. Yeah, that makes sense. So you sort of suspect that it's from humans and not from like pork. Do you have excuse me, access to any shedding data? There was a question about whether you have found any shedding data for HEV in feces or other excretions in the literature.
From the pigs.
Or human, or I think the question was for humans, but if anything would be interesting to hear about if you saw it.
So, uh, how much they excrete, or how long they excrete the infection. This is what
I
the
guess
question.
both. I guess both,
Okay.
like similar to what Krista and Abigail were talking about. Is there any data
Mm-hmm.
on hepatitis E on duration or quantity?
So they excrete in a very large amount, and if someone is infected, or if it's symptomatic or asymptomatic, they excrete till 4 to 6 weeks. And the chronic patients also contribute into the feces. So yes, it's there, and they excrete it for 4 to 6 weeks if they
Okay.
are infected. Yes, but uh,
Yeah, there's also a question related to that, which is given the like seems like long shedding and the fact that there are people that are asymptomatic
mhm.
that might be shedding, would you expect the hepatitis E RNA in wastewater to correlate to case data? I mean, you showed that it kind of, I mean, it didn't seem like it correlated that great, not the same way
Yeah,
like influenza, for example, would.
yeah.
So like, what are your thoughts on that? And then more generally, should we as researchers always be held to that? Because I do think if you submit a paper on wastewater concentrations, people do want to see that relationship show up.
Hmm.
So what are your thoughts on that?
So, so first, that we shouldn't compare two surveillance systems. So the clinical surveillance system to me is a separate surveillance system, and the wastewater is a separate. So we shouldn't expect that whatever clinical surveillance system is showing, that it should also show. It's an own a complement. It's a surveillance system. But for the diseases like asymptomatic diseases, where the data from the clinics and hospital are not enough to tell about the population level presence of the disease, there the wastewater can complement it. So it's for influenza, like for RSV, where you can already see the correlation and things, because they are also symptomatic, and then you try to do it. But for this hepatitis E virus, the most of the cases are asymptomatic. So then there is no, you cannot correlate it in any way. So here it's actually complement that system, not trying to show that it can work exactly like this. So here it's like both separate system, but they are complementing each other in a way.
Mhm. Yeah, I mean, I think that's a great response to that kind of question. Um, and And yeah, I think a lot of us on this webinar have experienced getting reviewer comments on papers about this. You know, like reviewers definitely want to see it correlated to case data, and then we have to explain why it doesn't. And it seems like we're always defending that, you know.
Yeah.
So it's great to hear your thoughts on that. Thank you. Okay, let's see. There are more questions here. Um, there's a question about sort of the diagnostic testing that's used for HAV. So I think we're still on this like clinical to wastewater comparison. So, you know, it says this question asker says that in most low income settings, the hepatitis E tests are serological and PCR
Mm.
capabilities might not be available. And also like maybe some clinics even in Europe or the US are using serological methods, using antibodies rather than PCR. And so I guess, how do you feel about comparing that? I mean, you already kind of explained how you feel like they're different surveillance systems, but do you have any
Mm.
thoughts specifically about that question?
Uh, so, so in Belgium, first, of course, they do the serological testing, but to confirm, they do the PCR as well. But again, this it's different from in the low and middle income countries to developing countries, because there maybe it's it's and from other way it's different genotypes, and also their system will be different. But here it's not where the eating contaminated water or something, so it's with the eating the undercooked or pork raw meat. And also most of the cases are asymptomatic. Only the people that are have that are immunocompromised, or that they have jaundice, but the first they got tested for hepatitis A virus, and then if it's not hepatitis A virus and they still have jaundice, then they go for the hepatitis E test. So 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 that's how they do here. But I'm not sure what she or he wants to ask about, like, is it this is the reason why we have less cases, the clinical cases,
I
because we
think
don't have yes.
I think it's just another thing to think about, like
Mm.
that relates to what we already talked about, which is like
Yeah.
it's really hard, like you said, it's really hard to compare. You wouldn't expect clinical surveillance to give the same answer as wastewater, and
Yeah.
this is another reason why.
Mm.
So okay, I think we have time for one other short question, which I think you already answered, which is about the duration of hepatitis E shedding relative to symptom onset. I think you mentioned this earlier, but can you
Yeah.
answer it one more time? Yeah.
So it's the duration for excretion is from 4 to 6 weeks if it's symptomatic, or also if it's asymptomatic.
Mhm. Thank you so much. All right. Well, we're out of time. Thank you so much to our speakers, to
Mhm.
Noor, Krista, Abigail. Thank you to Kyle. Have a great summer, everybody, and we will see you next fall. Thank you.
Thank you. Bye.
Bye.